Okay, so we are live and ready to go for our tutorial today. And this is going to be a very gentle introduction to principal component analysis in cytometry for dummies. So yeah, again, welcome to today's tutorial. My name is Ramji Srinivasan. If you are interested in playing along at home, you can play around with the demo data set at app.teiko.bio. So again, today's tutorial is about principal component analysis in cytometry for dummies like me. And this is going to be a very gentle introduction to a very powerful tool. And just as a reminder, our mission at Teiko is to make high parameter or high dimensional cytometry simple and easy for clinical trials. So who might care about today's topic? It's anybody who wants to find the essential drivers in a big cytometry data set. And this tutorial is heavily inspired by StatQuest and several others, so please like and subscribe to StatQuest channel. They do amazing work. In short, what it means is if you have a big matrix that is, say, 33 dimensions, 33 cell populations times 24 subjects, you can use magic like principal components to reduce this 33-dimensional data set to 2 dimensions and capture a lot of the variance. This is a preview of what we're going to walk through, but this is an example study comparing young, middle-aged, and centenarians and seeing the different immune profiles for these different patients. So how do we get here, and what does that all mean? What we're going to do is step through a synthetic data set inspired by real-life Lugli et al. In 2007. So this was an experiment design for this particular paper. They had 24 subjects, 9 young people, 8 middle-aged, 7 centenarians, and they captured 33 T-cell subsets from an 8-color flow panel. But you can imagine capturing hundreds, thousands, or millions, or potentially billions with unsupervised clustering. So we're going to show the synthetic data set, which is inspired by this, but then we'll also show the real-life findings to compare those. So for example, for each patient, they had a blood specimen, they ran it through a cytometer, and then they captured 33 populations, which are the rows over here, and then the columns are the individual subjects. So for example, the individual young people over here and centenarians on the right. And you can see here the percentages, which are the percentage of the immune state captured by this particular cell type that this person had, like 2%, 3%, etc. So these rows and columns are what we're going to do principal component analysis on. So let's step back a second here. So we said 33 populations by 24 subjects. If we just do 2 dimensions, so we take a blood specimen, put it in a cytometer, and just capture 2 populations, and we have a lot of data, it's easy to plot 2 dimensions on an XY plane. So let's go back over here. So let's take this young one, 75% and 2%. We can plot somebody like this on an XY plane because we're only capturing 2 dimensions. So young one over here, young 2 over here, centenarians over here, etc. And let's reiterate that this tutorial is only useful if you are interested in caring about the differences between different populations. If you don't care about the differences between, say, dose groups, response groups, then this probably isn't a useful tool right now. But if you do care about the differences, then principal component analysis is awesome. So let's bump it up to 1 more dimension. So if you went from 2 dimensions to 3 dimensions. So now we have individual cell populations over here, cell population 1, 2, 3, and the individual young people, and then centenarians on this side. At 3 dimensions, this is also not so bad. You can see the X, Y, and Z plane over here, and you can see the separation between centenarians and young people. So remember, in the X, Y, Z plane, we can plot all of these dimensions. And we can see this kind of clustering, and we can see this kind of separation. But what happens when we go to 4 dimensions, or 30 dimensions, or 298 dimensions? And this is where minds get blown. So the human brain can only visualize in 3 dimensions. And so this is where PCA, also known as principal component analysis, comes in. And there's going to be some fancy mathematical jargon, but we'll hopefully break it down. A reminder, PCA is a way to see the essential drivers in a big cytometry data set in 2 dimensions or 3 dimensions. So if you've got 40 dimensions or 100 dimensions or whatever, you want to reduce it down to 2 or 3 dimensions, but not lose a lot of the variation in the underlying data set. So this would be the end product, if you have a 33-dimensional data set, you'd ultimately be able to reduce it down to a 2-dimensional data set, as we showed in the beginning, with centenarians, middle-aged people, young people over here. You can see PC1 and PC2 are little variables here that we're going to show in more detail. And these have numbers here of the amount of variance in the data set they capture. So 24.18 over here and 12.23% variance. If you sum these up, these will constitute 36% of the variance. So we're going to walk through exactly how these are computed and what they mean for your data set analysis. So how did we get here? Well, we took this big matrix with 33 populations and 24 subjects. And remember, these 33 populations are dimensions, and these 24 subjects are rows. And then we computed what's called a covariance matrix, which is taking all these populations and comparing them to one another. So on this side of the matrix, you can see an excerpt. This is about 5 of these populations: CD4 positive, TN, CD127 positive, CD95 minus, CD38 dim minus. And we covary it with itself, which is how much does it covary with itself, and how much does it covary with every other population. So this is going to be what's called a symmetric matrix, a square matrix. And the diagonal values, so along the diagonal, these values are large values are going to be interesting, and small values are going to be likely to be noise. And the off diagonal can help us with redundancy. So large values are going to be redundant, and small values are going to be noise. So to build your intuition for what that might be, why that might be so, let's take a dummy data set. I'm going to show you the raw data for these populations. So if you look over here, this is subject 1 through subject 10, and sample cell populations over here. So large values on the diagonal are interesting, and small values are probably noise. So let's take the first population. This varies a lot as you look from left to right. So you start over here, it goes up and down and so on with the subjects. So this is very likely to be an important population and will have a lot of covariance with itself. This population labeled noise doesn't vary that much as you look left to right. So this population is likely to be noise. These two populations covary a lot together. So this is redundant 1 and 2. These guys go up and down together. So they are unlikely to be useful, and perhaps one of them can be dropped. And off diagonal, these vary a lot from one another. So they have a high covariance as you go from left to right. So remember, we have this 5 by 5 excerpt of a matrix. Now we can zoom out to all 33 populations and see the true covariance matrix. And again, across the diagonal, the markers that have a high covariance are, on the diagonal, the markers that have a high covariance with themselves are likely to be interesting. And if they have a close to 0 or low covariance on the off diagonals, those are also potentially interesting populations. So PCA takes these factors into account. So now we can compute the eigenvalues of this matrix. So eigenvalues are a very complex term, and we're not going to go into the entire derivation here. But from this big matrix, you get all the individual principal components here, and then individual eigenvalues. So eigenvalue 1 is 3024.8. That's 24% of the total variation. So this is computed by taking 3024 and summing this entire column together and getting to 24%. So this captures a lot of the variation in the population. Principal component 4, 573.7, this is 8.17%. So 573 divided by the entirety of this column gets you to 8.17%. So here's a definition here. The PC1 axis is the first principal direction along which the samples showed the largest variation. Let me pause again here. This is beyond the scope of this tutorial to explain eigenvalues and eigenvectors. There's a great tutorial linked here that you will be able to see in the YouTube, and that's from 3 Blue, 1 Brown. So if you're interested in that, feel free to check that out. Let's assume you have these principal components derived for your cytometry data set. So you've got PC1 through PC7, and you've got all these individual eigenvalues. Each principal component is going to correspond to a set of loadings. So let's take PC1 and this particular eigenvalue, and you're going to go down over here and you can see these coefficients are going to tell you which subsets matter the most. So for each cellular population, you're going to see PC1 loadings, negative 0.237, 0.195, 0.183, 0.307. And we're going to be able to plot XY from these PC1 and PC2 loadings. So we start here with this raw percentage, 75.4%, and these coefficients will tell you which subsets matter the most. So PC1 loadings, PC2 loadings, and we multiply through. So 75.4 times negative 0.237 gets you to 17.870. 75.4 times 0.156 gets you to 11.725. So this is what we're going to plot on the XY plane. So this is the PC1 and PC2 axes, and you can see the centenarians, the middle-aged, and young people all plotted. So we reduced 33 dimensions down to 2 dimensions by taking PC1 and PC2. So what is the value of this entire exercise? We've captured a 33-dimensional data set in 2 dimensions. So you've got a big cytometry data sets with dozens, hundreds, or thousands of cells measured by specimens, and you want to see what drives the difference between an endpoint, like response or dose. Now, bonus is to explain correlation circle plots. This is a way to graphically represent the relationship between the original variables and the principal components. So for these individual T cell populations, you're going to have the position of the variable on the plot, and the eigenvalues, which are going to describe the magnitude of variable contribution. So for example, PC1 loading of 0.29 is high, and then you've got 0.13 and 0.12, these contributions are quite low. So let's see how to read a correlation circle plot. So over here, you'll see similarly the PC1 and PC2 plots, and you'll see different cell populations. Let's take this one, CD4 positive, TN, CD127 positive, CD95 positive, CD3 dim. The magnitude, long arrows contribute more to the variation, and short arrows don't contribute that much. And if it's more horizontal, then it contributes more to PC1, and more vertical, it contributes more to PC2. So this is showing a correlation circle plot for the top 5 T cell population. So let's go to some real findings from Lugli et al. And this actually came from the real life data set. So you have young patients over here, middle-aged patients over here, centenarians, and their PC1s and PC2s and PC3s were much better than our synthetic. They had 60% variation in the first principal component, 28% over here, 4.77% here. So they captured a lot of the variance, like 87 plus almost like 91, 92% from all 3 components. So they were capturing a lot of the data set variation in their real life data set. And they found that young donors and advanced age were different based on memory T cells, and specifically these differentiation markers were up in young donors, and these differentiation markers were up in advanced age. So now let's turn to a Teiko data set. So we offer PCA on app.teiko.bio, and this came from University of Minnesota in 2022. This is a phase 2 clinical trial with 40 subjects, 20 specimens, 20 responders, and 20 non-responders. So let's click on over to see this data set, which you can also play around with. All the credentials are over here, so you can log in whenever you like. And we're going to go to the UMN Dataset here, and we're just going to quickly reorient ourselves. Remember, there were 40 samples, 20 subjects over here. And let's go to group similarity. And here you can see the principal component analysis and the explained variance, PC1, 55%, PC2, 19.6. So these do a good job of capturing the variance. So let's color by response so we can see a little bit of separation between responders and non-responders. And then further down, we're going to, let's shrink this down to 7 contributors so we can see different contributors. What is driving the difference? So for example, classical monocytes are driving a big difference. CD4 positive T central memory are driving the second most. And cytolytic NK cells, which one wouldn't necessarily expect on a classical flow panel, are the third here. So yeah, you can see that in a little bit more detail over here. So classical monocytes all the way down to cytokine producing NK cells. So this could tell you what drives the difference in treatment response as well. So wrapping up, PCA is particularly useful when the data set is highly correlated. So you can assume that there may be redundancy in the data, or that you can potentially drop variables without losing an explanatory power. So this is a great quote, which is PCA can be used to reduce original variables into a smaller number of new variables, but still explain a lot of the variance of the original variables. In fact, you can even quantify how much is retained afterwards. So with that, we'll wrap up. And if you're interested in more, you can check out app.teiko.bio. We have the data sets there that you can play with. And if you've got any further questions, feel free to email us or contact us at teiko.bio. Thanks for signing on.